Good afternoon, everyone. Thank you for joining us for this special lecture. I'm Debbie Ingalls. I'm the Director of CCOVI here at Brock University. And this afternoon, I'm pleased to present our speaker, Dr. Federico Tondini from AEB Biochemical. So Federico completed his PhD in wine science at the University of Adelaide, and he's just shared with me he's just doing his final touches up on that final thesis, so that's a huge undertaking. And his expertise are in microbiology, food biotechnology, and fermentation science. His main research areas include indigenous yeast, wine fermentation, and cell physiology. And as we all know, there's such a huge interest now worldwide on indigenous yeast fermentations and new strains of yeast and species of yeast coming into the marketplace. So we're always excited to hear about new applications. And today he's going to be speaking to us about yeast selection for cider production, because we have a huge cider industry growing here in Ontario and really across Canada. So we're really excited to have you join us today. So please join me in welcoming Federico.
Good afternoon, everybody. So as she said, the presentation today will focus on which yeast can I use to ferment cider. And this was a presentation slide, but yeah. Basically, I'm a biotechnologist student. I did my master in beer fermentation, studying why different yeast was producing different amount of diacetyl in lager beer strain. And after I moved to University of Adelaide to do my PhD in non-conventional yeast, non-Saccharomyces yeast. So I did some work on non-Saccharomyces and thiol. I sequenced the genome with this new technology, nanopore sequencing of Torula Brucki, and I tried to link this different genome to the enological properties of this yeast. This is still has to be approved. So at the beginning of a spontaneous cider fermentation, a mixed population of non-Saccharomyces yeast that lives on the apples and Saccharomyces that are fewer on the apple but usually live inside the winery are present in the juice. Natural fermentation usually gives more complexity to the final product, and they give that bouquet, aroma bouquet, that is described as terroir expression. However, this kind of fermentation are less predictable and less reliable. This because at the beginning and during fermentation, a lot of stresses the yeast have to overcome. This stress is the osmotic stress at the beginning, where the sugar are pushing into the cell, nutrient limitation during fermentation, especially when multiple species are present in the mast, and the ethanol toxicity at the end of fermentation. So it's very difficult to assess the risk of a natural fermentation, and it's very difficult to predict the outcome of this natural fermentation. So which yeast has been found in cider fermentation? A lot of non-Saccharomyces yeast belonging to the Methnicovia, Torulaspora, Candida, Pichia are present at the beginning of fermentation, but mainly two yeasts have been found in the middle and end of cider fermentation. And these yeasts are belonging to the Saccharomyces genus. Usually at mid-fermentation we can find Saccharomyces bayanus, while at the end of fermentation we can find Saccharomyces cerevisiae. So who are these yeasts? So Saccharomyces cerevisiae is the most used and most studied yeast on the planet. Saccharomyces cerevisiae is used mainly for beer, wine, and baker yeast. And it's become the most popular yeast because it has been studied in all the labs of the world because it's an eukaryotic cell, so very similar to mammalian cell. It's a simplified version of a mammalian cell. So in the genus Saccharomyces, there's the different species. There's the species Cerevisiae, but there are other 7 other different species with different characteristics. The ones that are more interesting for the food and beverage industry are Eubayanus, Uvarum, and one I could never pronounce it, Frugiazeri. And this because they are the most psychotropic species, so most resistant to cold temperature. So why Saccharomyces become so popular, so famous in food and fermented beverage? Because Saccharomyces has this particular metabolism, and that is called Crabtree effect. So the normal yeast eat sugar to make energy and grow. What Saccharomyces is doing is he eats more sugar that he can handle. A part of it is using to grow, and part of it is just producing ethanol. Why is producing ethanol? Because ethanol is toxic to other species. So while it's growing, it's producing ethanol. And this, inhibiting the growth of other microbes makes him like fitter in the environment, creating advantage in the environment. And humans use this characteristic to produce fermented food and beverages, because ethanol like prolongs shelf life, and it acts as an effervescent. So in nature, not only a pure Saccharomyces species has been found. What has been found in nature has been found hybrids of Saccharomyces. So where different species of Saccharomyces share their genome together. And the most famous are the lager yeast Saccharomyces pastorianus. It's a mix of 3 species. And there's other different hybrid. This one is used for Belgian triple beer, for example. And the other are all used for the wine, cider, and brewing industry. And the interesting thing on this hybrid is that they mix together the characteristic of their parent species. So for example, if we see the first graph, the hybrid is mixing together the ability of Saccharomyces bayanus to grow at a low temperature and to Saccharomyces cerevisiae to eat maltotriose. So it makes it fitter and better to grow in cold warm, but it's perfect for lager beer. And if you see, this hybrid has retained the optimal characteristic of each species to produce acetate and esters that are part of the aromas of the final product. So a little bit of history on the yeast. The yeast is the oldest industrial microorganism present that we know, and we're thinking they already started using it in the Neolithic, but the first representation of fermentation belongs to an ancient Egyptians 5,000 years ago. And what they used to do is that in the past they were using the natural microbes that were on the grain or on the grapes to ferment, and or in the air, and this yeast usually was like wild yeast and lactic acid bacteria. But on the course of time, bread after bread, if one kind of bread or one wine was better than the other, population was keeping a little part of it to restart the next one. And this brought to domestication of the yeast. If we define domestication, domestication is can be defined as like the selection, the human selection of a particular wild species that he became more fitter in a man-made environment with a man desire characteristic, but at the cost of the survival in nature. A good example of these are like dogs. Exactly the same. So people during the centuries, millennia, keep passing this starter from one production to another, keep selecting them, and keep improving their product. But nobody really understood what was going on inside. Until in the 17th century, for the first time, yeast cells were observed. In the 1785, the chemical reaction of fermentation was discovered, but nobody knows who was doing it yet. Until in 1857, Louis Pasteur finally demonstrated that fermentation were carried on by a living organism that was eating sugar, producing ethanol and CO2, and from there, yeast research rises. And related to food and beverage production, use of pure culture of yeast to improve the control, improve the reliability of the product has been employed until the 1880s in beer. And in wine, the first inoculation of a single culture was in 1890, but the industry didn't pick up the practice until 1970s. So, in cider, or traditional cider fermentation, it happens the same thing. So the apple was crushed, there was addition of SO2 to reduce at the beginning the bacteria contamination, but the fermentation was spontaneous and at very low temperature to reduce exactly the contamination of the yeast and let the fermented yeast produce ethanol. And once ethanol is produced, the bacteria could not thrive. But nowadays, again, to have a proper control and have faster fermentation, a clean and consistent fermentation, yeast in the cider industry is moving to a select inoculum of yeast. What's the problem of and a single yeast inoculum? Is that if in a natural fermentation there's a lot of yeast producing a lot of different compounds, in a single inoculate fermentation there's just one yeast that cannot produce everything. It can produce just single or like fewer molecules. So the debate is still open if it's better to have a cleaner, safer fermentation or to have a more complex aromas of flavor, but with a more risk. So it's a balance that the industry have to think about it. But today we are not going to talk about spontaneous fermentation, because every region, depending on the climate, depending on the apples, depending on each factors could influence like the microflora on an apple. So it's really like case studies. So it's very important to understand what domestication has done to the yeast. So domestication selected for particular characteristic of the yeast, desired characteristic. For example, beer yeast, they are being selected to not produce hop flavors, to be more able to eat longer sugar. In beer, the difference between beer and cider is cider has like glucose and fructose, but one maybe sucrose, that are one or two maximum sugar, while in beer you have like glucose, maltose, 2 sugar, maltodextrose, 3 sugar, and longer sugar. So in beer yeast, this yeast has been selected to eat faster this longer sugar. In wine yeast, the wine yeast has been selected for other characteristics, as to resist to more sugar at the beginning, as to resist to more ethanol, as to resist to SO2 that in beer are not used. So, and this yeast diversity really reflects human history. So, I just want to make one question today, and is, do you think is more diverse beer yeast or wine yeast? Just based wine. So if you think of wine making, how many times you make wine a year? 1. How many times you make beer? 352 weeks. So beer yeast has been selected 50 times more faster than wine yeast. And beer yeast is very strange because it never, so wine yeast you use it, and even if you live in the winery, as to survive a year without nutrients. While beer yeast, you take from a beer, you put in an oven, and another, it's always being feeded. So the diversity of beer yeast are like way higher than on wine yeast. And there's that group of mixed yeast that, yeah, have unique profiles. So, and you can see that the different industries really shape the yeast, the yeast we use. So, what yeast do during cider fermentation is basically, at the beginning, the life phase has to face this hostile environment. Once you understand that there are nutrients around, you start growing. Usually inoculation in wine and cider are 5, 10 to the 6. And while in beer you tend to go a little bit higher, 10 to the 7. So as soon as they understand that there's a lot of food, they start fermenting the juice, so converting sugar to alcohol, until the environment becomes too toxic, and so they stop their growth, but they keep fermenting until they finish the nutrients, and so they start dying because of the high concentration of ethanol. This is a pretty easy understanding of alcoholic fermentation. So from a molecule of glucose, yeast produce 2 molecules of CO2 and 2 molecules of ethanol, producing energy and restoring the cofactors needed. So what's important to understand is that yeast, the main 2 nutrients for yeast are carbon, so sugar, and nitrogen, so like amino acid, but there are like the building block and the energy of the cell, but all the vitamins and trace elements and minerals are the gears of the cell, that without that the yeast cell cannot function. So the yeast use these nutrients and materials for growth and fermentation. So on the side of these main metabolic pathways, one is the energy pathway, and this energy is used to grow. Why the yeast cell produce these flavor molecules? So if we go and see what the biochemistry inside the cell, papers show that these molecules are produced to balance the metabolism inside the cell. So it's all about chemical reaction, that if they go in one direction and you want to keep in one direction, you have to restore the intracellular balance. Or to detoxify. You're producing too many of these compounds to do another things, and it's becoming toxic. So what's the best thing? I just produce an ester and it goes away in the air. However, what's the interesting thing is that a lot of these flavor active molecules are produced by the means to communicate. So some of them are used to communicate with each other, some of them are used to communicate with other species, even bacteria. But one of the most interesting things that has been published is that these esters have been proved that are to attract flies, mosquitoes. So what's the advantage of this? So you're on a rotten fruit, like a broken fruit, and you're eating your sugars, but that is not going to last forever. So what do you do is to attract flies that go from one to another, that picks you up and bring you to another fruit. So, and clearly, so all these flavor-active compounds, there's 100 in fermented beverage that can modify the aroma profile. And the major attributes are usually referred to yeast fermentation. And these are mainly alcohols, higher alcohols, ester, and carbonyl compounds. And there's a need to understand how to produce these compounds in higher quantities, because it's a key for success. Because I think winemaker, brewer, cider maker, he wants to, one product is not better than the other one. They're different. So if you want to create a new product, a distinguished product, and you want to produce more of a flavor, that's why the industry wants to understand how to produce this flavor. And and that the market is going right now more than quality of the product, more than the quantity of the product. So if you can produce a better quality product, you are a major chance of success. So how yeast could influence the fermentation are like these 7. So it can influence at the beginning. It's called, it's very popular now in France. It's called like a bioprotection. so where you spray yeast on the grapes already to like avoid molds and other things to grow. Again, with its metabolism, producing de novo compounds during fermentation, with enzyme production. So there's a lot of flavors that are bind to compounds in grapes. So they need to be cut it off and released to be perceived. Autolysis, so when the yeast die and you keep your wine or cider on the lees, it can realize an amino acid can have a savory or sweet taste. It can absorb juice components. Some yeast could be spoilage. Like for example, there's a big controversy like in beer, Brettanomyces. It seems like a maybe good yeast to play with. In wine, no. I don't know in cider. Maybe you can enlighten me. No, good. And yeah, and again, because of its production of ethanol and interaction with other spoilage microorganisms, it could like bacteria. So what we're focusing now today is the de novo biosynthesis of products. So when yeast can thrive in the environment, has been shown here with like grapes, because the major of research has been made in grapes and beer. It can ferment, produce aromas with secondary metabolites, could produce enzymes that freeze juice molecules. And what research have done, applied research, is how different yeast with different genomes can produce wine with completely different profile, aroma profile. And since the 1996, where the first yeast genome has been completely sequenced, research have an in-depth look at the phenotypic characteristic and the genetic characteristic. And this brought to the understanding of the genes involved in these esters and aroma production. Especially now we can see that, sorry for the blurry image, but higher alcohol, acetate ester, and ethyl ester can be reconducted to amino acid metabolism, fermentation of sugar, and lipids metabolism. And the genes have been detected. How have been detected? We know the genome, we knock out the gene, or we overexpress the gene, we measure the production, and we can see if they are involved or not. So basically, higher alcohol are produced from amino acids through the Ehrlich pathway. So the yeast cell takes the amino acid, but maybe it doesn't need that amino acid. It needs another amino acid. So you take out, and maybe it needs only the nitrogen. So you take out the nitrogen, you remain with the skeleton of the amino acid. It doesn't know what to do about, so I want to get rid of it. You produce first an aldehyde, and after an higher alcohol. Acetate ester, this is very short, we can get more in biochemistry if you like, but acetyl-CoA comes from the fermentation of the sugar. These are our higher alcohol before the yeast with ATF1, with esterase, it produces the ethyl esters, while acetate ester, while the ethyl ester are produced from intermediates of a lipid synthesis. During the growth, yeast is building its lipids for the membranes. They have to belong as much as like 20 carbons, but it's a reaction that needs oxygen. So a lot of time, yeast gets stuck with a lot of intermediate compounds. It doesn't know what to do, or it needs something to restore its redox balance inside the cell. So it combines a molecule of ethanol and lipids, and it produces ethyl esters. Ethyl esters are really, there's thousands of compounds that cannot be made. And each yeast has different characteristics, because each yeast prefers to produce one ester on the other ester, depending on its genome, on the condition, on the juice is fermenting. Another compounds that is very interesting are the phenols. These are very for beer, very interesting for beer, and for wine too. Why? Because usually Saccharomyces has been selected to stop here. So it can produce only the vinyl phenol. The other reaction is famous because it's Brettanomyces to produce it. But in wine and in beer, it's very interesting, with spicy notes, and like Belgian beer. So there's two ways that the industry are trying to get to bust, to obtain the desired flavor when it's fermenting. One is to adjust the fermentation environment, and one is to select the yeast strain with the genetic characteristic that you want. So are both big tools in the hands of winemaker, cider maker, but definitely the more variety and the more control is with the genetic, the selection of the desired yeast. So what science suggested to do was to create super yeast with desired characteristic. Oh, I want a lot of glycerol. I modify the pathway genetically to produce more glycerol. I want more ester. I put another copy of the enzyme, produce double esters. Unfortunately, it's not accepted by consumers. So one yeast, I think, in the US has been accepted to be GMO. So, because we cannot modify the genome of the yeast, how can we find the yeast with a proper genetic kit that is interesting for us? First thing is natural diversity. There's plenty of yeast out there, and with different characteristics. Another thing is mutagenesis. So it's basically it's like a non-GMO technique to modify the genome of the yeast. Basically, you recreate the yeast under the sun, and you wait that the light make damage in the DNA, random mutation, and after you see that if that mutation affects the flavors. Evolutionary engineering. So you put the yeast in a super stressful environment that you want. For example, you want a yeast that ferment 300 grams, 400 grams of sugar. So you put the yeast in these 400 grams, 400 grams, 400 grams. And so it divides. And the idea is that the fitter, the better, will survive. So you make nature faster, again, by the mutagenesis. Or sexual hybridization is what happened in nature, what happens in beer, for example, in the beer yeast, but you do it in the lab. So you don't have to wait for 1,000 years of brewing to get your desired yeast. So, as I said before, making an hybrid is like what has been done for for the modern dogs, all modern dogs came from the wolf. But we selected the desired gene for the desired characteristic we wanted until we had like the phenotype express that characteristic. And it's the same. The yeast usually have like 2 chromosomes, so easy. Basically genetics. One will be AA, and you will have all the possible combination, and after you have to screen for the one that have your desired characteristic. So this is a paper published 2016, 17, I think, where we create a new hybrid to perform, to have a pleasant aroma profile and high tolerance to low temperature. Here wasn't really an hybrid. Here was more a screening, natural screening of the yeast, on which yeast was better for sparkling cider production. So the characteristic they were looking for was resist to ethanol and resist to 200 ppm of SO2. So my company did some research that I think could be interesting for the cider and wine. This was made in wine, but I think they're going to be very interesting for the cider perspective. One, this yeast is called Fermol elegans, and it doesn't produce sulfur compounds. And another one is called Fermol Glutathione 1. Basically, this yeast, it produces more glutathione. Glutathione has a reductive power, so it can protect the aromas from oxidation. So it protects the aroma during fermentation. And these are the 2 papers. And one was based on, again, evolution, non-genetically strategy, that basically impair the sulfate assimilation pathway. And the other one was made with evolutionary methods where there was like a metal that was like binding for the glutathione. So the yeast, and glutathione is very important for yeast to survive. So the yeast, to survive to the presence of this metal, have to boost the glutathione production, and they select the yeast with more of this glutathione production. Another one is, and this one is based for cider, is a Baianus yeast strain. It can enhance fruity, can tolerate ethanol, no H2S production, and it's cold and SO2 tolerant. How we did it? Basically, we checked all these parameters during the research. It has been used like this machine that can make 96 fermentation in the same times robotically. It's all high throughput, so I want to sample 3 times a day. Just put the robot taking the samples, putting in a 96 well plate in the ice. And when I have this 96 well plate in the ice, I have all the instruments to reduce the human impact on the research. Spectrophotometer for like old plate. We have a flow cytometer to measure the cell count, and we have a chem well to do sugar and enzymatic assay of what we want to check. So, and because they're made in glass vials, we can do aromas GCMS analysis at the end of it. So so what we monitor, it was different condition, but basically what we monitor and what you're interested in monitoring when you're selecting a yeast is how the fermentation goes, and how these are like two different yeasts. They were like trial, and you can see the fermol S2 as an increased ester and aromas production. But as I said before, another chance is to increase your diversity. It's not just to look at Saccharomyces, but is to use other yeast strains that usually are in spontaneous fermentation. So what biotechnology industry is doing now is to try to find non-Saccharomyces yeast to make innovative products, alternative flavors, and other kind of particularities in your product. For example, there's this yeast, CCOVI Lecture. It's basically used for bioprotection, but has been shown that it produces a lot of extracellular enzymes. So in sequential inoculation with Cerevisiae could enhance the apple variety flavor. And there's like a technique that has been tried in wine where like sequential inoculation reduces the acetic acid. Not sure how big acetic acid problem is in cider. Another one is La Chance thermotolerance. And they have different ethanol tolerance, these ones. It's an important thing you have to consider when you use this yeast. So CCOVI Lecture is only 3% ethanol, so you cannot leave it there for a week. You just leave it there 2 days, and after you have to put your Cerevisiae in. La Chance thermotolerance 7%. I think it would be very nice to produce this cider with this as a different ester production than Saccharomyces. And the interesting thing of this yeast is that it produces ethanol, but it produces lactic acid too. So you can make a sour beer or reduce the acidity of wine. And another one is Torvaspora del Bruchi, which has the highest non-Saccharomyces ethanol tolerance. Again, different ester production, if you want to understand. But different ester production based on different amino acid assimilation. It produces extracellular enzyme. It has been shown it increased thiols release in wine because of these extracellular enzymes. Produce more monoprotein and less acetic acid. So this is in our paper, but I think 2014, 16, not that new, but they tried different non-Saccharomyces yeast to improve the aroma and flavor of cider. So how to assess? Okay, so how do I choose? I have to make some cider. How do I choose the yeast for the cider? So first thing, you can go. This is the page of my company. You can go on a page on any company, and usually the cider-related product means that they did all the research job that I showed you before. So usually you can have like this job. So if you have like a cider part and the company is doing cider, it means that they're being tested in cider. So you can see which flavors in cider can express. You have to consider that it's a standard cider. It will never be exactly the same, but it's an indication. And you can see the production of SO2. You can see how much malic acid is consuming. I can't read that, but yeah, the production of H2S, how much nutrients they need, and how much fast they ferment. So if you go and you want to select a yeast from there, and you can read this, you can get an idea of what you want. But if you want to open and have like a different view on what to do, if you want to have an innovative product. So there's not a recipe. I'm not here to say, oh, you have to use this or you have to use this. I think you have to understand that what your raw materials, what's your finishing materials, how you're going to process, and what you want, in which direction you want your yeast to go. So you have to have an understanding of these parameters to choose the yeast. And it's all a balance. It's all about to you, to what you want to achieve. So first thing I want to suggest you is like to point out the differences. So, okay, I don't want to use a cider yeast because they already used too much. I want to use this beer yeast. Okay, beer yeast has a lot of selection with a lot of different flavors that you can have. What you have to understand is what beer wort has that cider apple juice doesn't. So I think the two main differences are between a beer and apple are the sugar inside. So the total amount of sugar are the same, but in beer are mostly maltose, maltotriose, glucose, not a lot of fructose. Like, while in the apple is the opposite. There's a lot of fructose, and there's not a lot of glucose, and there's no complex sugar. So, in fructose, yeast can metabolize it, but usually when it reaches a certain level of ethanol, it might just stop fermenting. So, using a beer yeast could produce you like different flavors, could then have a lower attenuation of your cider. If you're selecting a wine yeast, it's good, but I think in wine there's a way more amino acid, and in beer too. In cider, I think you have to select for a yeast that have low nutrient requirements, or you have to supplement a lot of nutrients, organic complex nutrients, and not that. So first things to choose is understanding what you want to ferment, and this can give you an idea on which yeast you can select. Here I just take the opportunity because apple juice have I think a problem of H2S production, which depends because of the low yeast as young. And last week I were in Australia mounting this machine. Basically you put it on the top of your fermenter and it can monitor CO2, the blue line, and H2S production, the red line. So as soon as you see, and you can send it to your cell phone. So as soon as you see H2S, okay, put nutrients. And you, instead of solving the problem at the end, maybe with copper sulfate that is ruining your even the flavors at the end, fruity flavor, you prevent it at the beginning. Again, when you choose your yeast, you have to think about your process, because there's a lot of ways to affect this aroma production. If you want it more neutral, if you want it more fruity. So usually, if you oxygenate your, I don't know if in cider you have this practice, but in beer you oxygenate your wort. More oxygen means less stress cell, lower ester production. More stress cell, more ester production. And the pitching rate is the same. If you pitch more yeast cell, they actually divide less, so less stress, less ester production, low pitching rate, more work for one cell, so more stress, more esters. Another thing that you know is temperature. High temperature results in higher ester production, but I think it's like a machine like going fast. It doesn't care what it's producing. It's producing everything, so you get a lot of everything. While with lower temperature, you will get less ester production, but sometimes less is more, less is better. So you can have lower ester production, but with a better final aroma. And another thing is sugar and fat, especially the nitrogen. Nowadays, have been developed complex nutrients that can boost your aromas, and some amino acids have been related to ester production. So really, nowadays you have the tools to boost the particular aromas that you want. So very easily, maybe you're not happy of the yeast that you find online. So you want to, but you made this awesome cider last summer. So you want to collect your sample. It's pretty easy. You put some of your apples in YPD. That is yeast extract 1%, bacteria peptone 2%, glucose 2%. You shake it with like a flame or under a fume hood because you're in university. You streak it on a agar YPD. Agar YPD is like 2% agar in here. And you will have single colonies of yeast. And after you can score like with smelling or sniffing approach. Very easy. You can use your apple juice, 20 grams per liter of agar. So I think it's 2% of agar in your apple juice, and you can put it on a plate, and you take a single colony and you streak it on the plate. And that colony will grow. After a week, you can smell. If it smells good. If it smells bad, it was in the bin. And, you can go laboratory scale, 1.5 liters. And, it's very easy and very cheap nowadays to get your species identified. I think it costs $20, maybe a little bit more if you just do one, but it's pretty easy. And keep your yeast, because it's not good when you do good things and you can recreate it. Glycerol stock is pretty easy to do. You have to make 80% glycerol solution with water, sterilize it, and when you want to put in the freezer, just half of your yeast culture, half of 80% sterile glycerol, it goes in the freezer. Even minus 20 is gonna live for a while. So that's it. If you have any question, I'm happy to answer. And that's my email. I'm always happy to be involved. And I don't have answer of all the questions, but I think my part of my job is try to reach and take the information and try to explain and have some answer. Thank you very much.
We've got some time for questions. Does anybody have questions? Two things. One is, do you have a yeast for sparkling wine that produces more amino acids? Or even for sparkling ciders for beer? Is there a yeast that has an increase, releases more amino acids?
So usually the release of amino acid is for autolysis. So when the yeast cells dies. So we have a product for second fermentation that can create that.
What's that called?
I'd have to check.
Thank you. And is there a particular yeast, have you found, I don't know whether you've done that, there's been a lot of studies on Maillard reaction compounds in beer and cider with the caramel toasty flavours. Is there a particular yeast that can produce, that speeds up that reaction at all? Or that is involved in that reaction, it's other than the...
So which compound again, sorry?
The Maillard reaction compound, so the caramel toasty flavours that it recycles.
Right now, I don't know.
Okay.
I don't know. I can look about it. If it would be easy if we know in which product are used, so we can understand which yeast are using, and to understand how that's happening. Usually, I know like in wine, that sort of flavors maybe are given with another, given by oak usually, or tannins. But caramel.
No, but that's fine. That's fine. That's fine. Thank you. I'll email it. Thank you very much.
For secondary bottle fermentation, I've used 3 different types of yeast, and after aging I haven't taken it out when I haven't aged it, but it's been aged for no less than 15 months. I've had a lot of tannin haze, or chill haze that you get, and it's not in the baseline for the base side prior to the bottle fermentation. Do you have any idea on how to avoid that or deal with that?
So, based on do you think it's the yeast that is causing that, or is just because of the changing of the condition in the bottle that is creating a protein instabilities? Because
it's only on the secondary fermentation bottle. I've used Spark, I've used DD10 and EC11.
Yeah. So I think it's because it's more the yeast and the bottling conditioning are changing the composition inside of the bottle. So in my opinion, you can try to protein stabilize differently before.
It's only when it's chilled. So you chill it to 1 degree, it gets haze.
Yeah.
You bring it to room temperature, it's clear.
Yeah, it's protein stability. Yeah.
Did you say that during the secondary fermentation?
We can discuss.
Any other questions? Just one from me. Just with some of the non-Saccharomyces cerevisiae strains that are coming onto the market, are you finding winemakers are excited and gravitating towards these new yeasts to trial out to still try to get that complexity without that same level of risk? If they were doing a spontaneous ferment, or are they still a little slow to, adapt these new products?
It depends on the size of the winery, and so there's. But usually, if the person that use non-Saccharomyces are already using spontaneous fermentation, so there's not a lot of winemakers that are using non-Saccharomyces yeast inoculant. What is interesting is now non-Saccharomyces are using more for other purposes, more than fermentation. Now is bioprotection is becoming a big thing, and I think more than on non-Saccharomyces yeast, wine world is moving on into like organic, biologic. I don't know how to refer it, but so they want to have more natural thing to use. So and a lot of this yeast produced the researchers saying that. Have positive effect. Like one, for example, the one that drop the pH, reduce acid addition, Pulcherima produce this acid that inhibits bacterial growth, or it's sequestering iron from the media, so it's stopping our spoiled yeast to grow. For fermenting, I think they still prefer Saccharomyces because it gets the job done. Yeah.
Very good. Well, thank you very much for a wonderful talk today. And here's a small token of our. And please join us next week for our own Dr. Belinda Kemp, who's going to talk about communicating scientific research and knowledge to grape growers and winemakers. So, what are the tools that we can use to improve our communication? So please join us next week for a really exciting talk. Good to see you all. Thanks.